All right. My name's Jerry Feidelson. I'm the CEO and co-founder of Agribody Technologies. We're working on a problem very similar to what was described, which is food waste. And I think it's pretty clear, if I can advance my slide, that we have enormous losses. Almost a third of the food is lost. It varies by crop type. About $1 trillion a year is what Xander mentioned already, mostly in the developed world, some in the emerging countries. In addition, when food is wasted, there are a lot of secondary effects like greenhouse gas emissions, particularly methane. So if food waste was a country, it would be the third most polluting country in the world after China and the United States and ahead of India and Russia. The UN has a major sustainability goal to cut food waste in half in the next 10 years, which is really quite ambitious. So the current methods are based on physics and chemistry, temperature control, chilling fruits and vegetables and other perishable products obviously works, but that has a lot of CapEx and OpEx. You can eliminate gases or try to reduce oxygen, increase CO2 or nitrogen to try to keep produce alive longer. We already heard a talk about edible coatings. And what we do is different. We work inside the crop. There is no OpEx. We simply knock down a gene using genome editing, not transgenics, that controls a switch, an analog switch that causes crops to decay. We do it once for each variety, and all of the progeny have that trait of long shelf life, so there's no need for continued treatments. Obviously, this is really important during pandemics. Our products will not be launched in time, I think, for the end of COVID-19, but we certainly want to be prepared for the next ones. And I believe every seed company worth their salt is going to be breeding for longer shelf life for just these reasons. The way our switch works in a fairly non-technical way is illustrated by this cartoon. It's a little bit embarrassing that I have a PhD in genetics from Stanford and I have to show all of my science on one slide and it's a cartoon. But basically, you've got these two genes that work together. This gene modifies the product of that gene, and the cell is always sensing the ratio between the modified form and the unmodified form. When there's a predominance of the post-translationally modified protein, the cell goes into senescence and dies. So what my co-founder, John Thompson, discovered some time ago was that if he reduced the amount of this modifying protein, or alternatively, he increased the amount of its target. In both cases, in about a dozen different crops, all kinds of crops, monocots, dicots, annuals, perennials, the result of pushing this analog switch a little bit in favor of continued growth and a little bit away from senescence resulted in extended shelf life, 3 to 4-fold in a number of crops, also increased biomass and seed yield in row crops, as well as more tolerance to stress, both abiotic stress like drought, high temperature, low nutrients, as well as some diseases. Here's some pictures of tomatoes that have been, in this case, these were genetically engineered to reduce the amount of that DHS protein. And you can see the 7-week-old tomatoes are as good as the 1-week-old control. Same thing with bananas. This was done in a field trial by an Israeli biotech company. This top row reminds me of bananas I buy at Costco. They're either over the hill or underripe. We've got 15 issued patents, including foreign counterparts. We've got applications in process, including international filings, genome editing applications. We've in-licensed the non-CRISPR-Cas9 method from Benson Hill. So anybody that messes with our proteins are infringing our patents. We got a great team. I hired Venki from Illumina. We've got a very good CFO and lawyer. Also a number of advisors that I don't have time to go into, but we make money through conventional licensing or co-development. We're trying to raise money to build a small lab to do trait introgression. Ultimately, we might become a seed company. We're operating in a sort of a virtual mode like a specialty pharma company. And so we're able to accomplish a lot with very little funding. We've already raised 1.6 million in notes. We've demonstrated proof of concept in all of these crops. We filed our patents internationally, and we've generated about $350,000 in revenues. These are our customers so far. I hope in the next time we talk, there'll be a couple of more arrows, including strawberries. JR Simplot is in their 3rd year of field trials now. in potatoes, and they tell me if they can get a 10% yield increase, as they've seen thus far, they can increase their acreage from 350,000 to 500,000 acres. And at a $10 an acre royalty, that's a pretty nice annuity. We opened a $2 million pre-Series A round, and we have a very good idea of how to spend that money. We want to develop our own tomato variety that's long shelf life that we can use as a universal donor to breed into a number of other tomato varieties, etc. We hope to sell in 5 or 6 years. It will not be an IPO. It'll be a strategic trade sale, probably to one of our customers. We have a clear path to being profitable and to get significant revenues on a high margin. We expect to do one more fundraising, a Series A, in 2 or 3 years, and expect by the time that money runs out to be profitable. So I don't expect we'll have to do any additional fundraising after that. I'm driving around San Diego with this license plate, so this company has to be successful. So thank you very much. I hope I stayed within my time. I'd be glad to take questions.
Jerry, this is CRISPR technology?
Yes, it is.
Okay.
The target validation was all done using antisense methods or upregulation, but we started the company to apply CRISPR. And as I said, we have a license from another company, and we'll get licenses for Cas9 from Corteva.
Okay, and I love the technology because I think it's obviously far superior to some of the things we've been doing with GMOs, etc. But clearly there are some regulatory hurdles in Europe. I don't know what your thoughts are on that.
Indeed, you're right. It was very disappointing in July 2 years ago when the European Court of Justice went against the advice of their own chief lawyer to basically consider genome edits to have a single base or very small deletion to be regulated like transgenics. I mean, that scientifically is completely insane, but it's reality. And they also influence Africa and Asia and other countries. The fact is we still have at least 10 countries that are very open to this technology besides the US and Canada. There's also Brazil, Uruguay, Argentina, in Israel, Sweden, Japan, and Australia. So we have plenty of markets, more than we can deal with in the next few years. So I'm concerned about Europe. They've consigned themselves to essentially be the Museum of Agriculture, unfortunately. But we have great markets in high value produce and as well as horticulture and row crops. I don't know if that answers your question, Aidan, but hopefully Europe will get real. Probably the best thing about Brexit is that the UK can regain rationality.
Jerry, you're a lot more optimistic than I am. Congratulations on a great presentation. I'm interested in your aspirations in Southeast Asia and in China particularly.
Yeah, China is interesting. I've been to Thailand twice, pretty much giving this pitch at fairly high levels. The issue in Thailand is that they have a very strong sense of organic agriculture. So it's not unlike Europe. China, again, is very picky in what they decide to ban. So you can't bring in transgenic alfalfa, but it's fine to bring in transgenic soy and corn. So again, it's probably more market protection than anything else. Yeah. But we'd like to find a partner in those countries. It's sort of outside of our reach right now with the staffing we have. So we're focusing on the US and, well, North America.
Okay, thanks so much, Jerry.
I have one question about your time to market. What is your time to market looks like, particularly in North America?
Right, so this requires patient investors because you can't rush plants. Plant breeding is something that just takes time. There's no way around that. So what I say is that by using genome editing in a rapidly growing annual crop like tomatoes or many other vegetables and flowers, it adds 1 year to the product development timeline of what it normally takes a new variety to be released to the market. So if it takes 4 years to get a new tomato variety through the entire system to prove its trade, to show it's safe, that it's agronomically valuable, it would be 5 years with our technology. And that timeline will shorten once we can breed it in through conventional breeding.
Okay, great. Thank you so much, Jerry. That's a great presentation. Very interesting technology. I'm in the middle. I'm trying to find out, should I be optimistic or should I be conservative?
Right. Well, we're just looking for someone that can write a half